can go ahead, ma'am.
Thank you, Dr. Ravi, and thank you, Novakum, for the opportunity given, and thank you for the kind introduction. So with this, I will just start my presentation. So I once again welcome you all for this presentation on bacterial fish pathogen, their isolation, identification, and management. So there are several bacteria which affects the fish. And so if you classify some of them, the major ones being Aeromonas salmonosidae, which is very common in salmon, not widely reported in India. And Aeromonas hydrophila, yes, which is a causative agent for hemorrhagic septicemia or red disease, and which is commonly reported in India. Edwardsiella, of course, there are some literature which has been isolated in India. Yersinia, Flavobacterium columnar, which is also responsible for columnaris disease and has been reported in some literature. And Angliseptica are also reported in India. And Vibrio, again, Vibrio is one of the pathogens present in fishes. And others, Streptococcus is very big problem in tilapia. And so these are the commonly reported bacterial pathogens in fish. And out of which the major concern is of Aeromonas and Pseudomonas and Vibrio to a certain extent. Vibrio is more a halophilic bacteria. That means it lives in salt. So in freshwater fishes, it is Aeromonas and Pseudomonas and Edwardsiella to a certain extent and Flavobacterium. So we will concentrate on some of the major bacteria which has been reported in India. So first, let me talk about Aeromonas. So the commonly found species in India are Hydrophila and Veronae. Aeromonas hydrophila and Aeromonas veronae. Salmonosida, of course, we've got few strains, but this is more common in salmon. So what is the problem which is caused by Aeromonas hydrophila? So the disease is called as hemorrhagic septicemia or also red disease or red sore disease. So when you say septicemia, it's an infection which is affecting the blood and the internal organs. The entire animal is, the systemic infection is happening in the animal. That is septicemia. And second is fin rot, where there is degeneration of the fins. And it is also causing ulcers on the skin and the fish. So it's also called as ulcerative disease. So what is the symptom? So when Aeromonas is there, there is sudden death in otherwise healthy fish, lack of appetite, there are swimming abnormalities and pale gills, and the animal has a bloated appearance, which is called as dropsy. You know, you have an abdomen which is bloated on either side, and skin ulcers are very commonly reported. These ulcers are commonly surrounded by, let me just show. So it's surrounded by a bright rim of red tissue around the ulcers. So this is very common in the Aeromonas infection ulcers. And as I told you, it's a systemic infection. So it affects all the internal organs, which could be kidney, gills, liver, spleen, pancreas, and even the muscles. So this is a notorious pathogen in fish culture today, causing major problems with the pisciculture. So the next important notorious pathogen is Pseudomonas. And here it is, in India, we have isolated a lot of Pseudomonas fluorescens. So this is also having symptoms which is very similar to Aeromonas. First is septicemia, which affects the blood and internal organs. And again, this also causes fin and tail rot. So there is a degeneration of the fins and the tails of the animal. And this also causes the skin and gill infection, very similar to the ulcers which we have observed in the Aeromonas. So this is more or less similar symptoms between the Aeromonas and Pseudomonas which is observed. So what does one do if you have to isolate the bacteria and identify from the diseased fish? So the first step, what we do is normally to get the diseased fish and collect the representative sample, which could be the internal organs or the ulcerative tissue. Normally, we don't take in the ulcer, which is we go little away from the, slit, the healthy tissue just adjacent to the ulcer, or it could be gills, or it could be kidney, or it could be any other internal organs. So we collect the sample and make sure that the container what we collect is sterile, and we wash the sample very well so that we don't have contamination which is coming from the handling or from the water which is there in the pond. So we make sure that we wash the fish very well and collect the respective samples in the sterile container, and you take it to the lab in cold condition. Normally, we don't ship it in the room temperature because if it takes 24 to 48 hours, there is a good chance that other bacteria is going to multiply in plenty. So very important thing is to collect appropriate sample and then ship it to the lab in cold condition. Then the next step, what we do is to isolate a single type of bacteria. So which is done in the following slides, I will explain about it. Then the next part is to identify the bacteria based on the biochemical characterization. And the next step, biochemical characterization is not always a conclusive, test. So we always go for molecular characterization in the next step. Once you have characterized and you have a strain which is, a single type, then we preserve the strains by lyophilization. So these are the steps which are involved in the isolation and identification of any bacteria. It could be a fish or it could be any other animal. So what do we do for isolation? So normally bacteria is isolated either in an enriched media or in a selective agar. So supposing if you are shortlisting, I want to isolate only Aeromonas from these fishes. So you are very suspicious that these are, infection of Aeromonas, then we go for selective agar. Or if you are sure that this animal may have Aeromonas and Pseudomonas, then we go for two selective agar. But if you are not aware what is the infection and you want to identify the cause of the infection, then we go for enriched media, which could be, say, tryptic soya agar or LB agar, which is Luria betani agar or nutrient agar. So we go for such general media if you don't know the cause of the disease and you just want to isolate the bacteria which is involved. So in this case, since we are going to go for, say, I'm just taking an example of Aeromonas. So if you're suspecting Aeromonas, then we go for selective agar. Selective agars are the one which will eliminate the other bacteria and selectively favor the growth of a particular bacteria. So if you are taking, these are available with the companies like High Media and other, media companies, where you can buy the selective medium in which only Aeromonas grows or selective medium in which only Pseudomonas grows. And many of these bacteria, say Aeromonas, is resistant to multiple antibiotic, which is a pathetic scenario today, which I'll come to it later. Many of the Aeromonas, which is isolated from fish or the deceased fish, are resistant to several antibiotic. It could be penicillin, ampicillin, novobiacin, etc., etc. So what we do is we take the selective agar and we add a particular type of antibiotic, which could be novobiacin or penicillin or ampicillin at a particular concentration. So this will favor the growth of Aeromonas selectively. So you could see that the first plate here, this is Aeromonas selective agar, where we have plated with antibiotics. So you can see that it is almost a single type of bacteria. So the Aeromonas grow in a yellow colored colonies on selective agar. So we pick up the single colony. This in turn is inoculated into LB broth or TSP. TSP is tryptic soya broth for further characterization. So if it is Pseudomonas, say for example, we use agars like Pseudomonas isolation agar, which is selective media for Pseudomonas, in which we add antibiotic like triclosans and other selective antibiotic like nitrofurantoin, which will inhibit the growth of other bacteria and selectively favor the growth of Pseudomonas. And again, after overnight growth, we pick up the single colonies and we inoculate into the other broth, which could be LB broth or TSB for further biochemical characterization. So this is the first step. And also when we are, plating this, you need to see that you plate appropriate dilution of the sample so that you get single isolated colonies. So this also has to be standardized and by your repeated trial and error. So you know at what dilution you will get single type of colonies. And this is also one of the steps. So this could be discussed a little later also, but anyway. So say, for example, if you're suspecting your organism is Aeromonas hydrophila, then we can plate it on what is called as blood agar, which contains sheep blood. So we see a zone of beta hemolysis. You can see that this is the place where we have streaked Aeromonas. So you can see a zone of wide zone of clearance of the hemoglobin. So this is called as beta hemolysis, which is a characteristic of Aeromonas hydrophila. And this is an agar in which Pseudomonas is plated. So Pseudomonas fluorescens has a character of glowing under the UV light. So you can see that the fluorescence, which is greenish blue in color, which is seen in the UV light. So these are some of the characters by which you can, boil down that this could be Aeromonas hydrophila and this could be Pseudomonas fluorescens. Okay. Now we have the plate in which you plated your organisms and you have single type which is inoculated into the LB or TSB broth. So now you have different colonies which are present as 5 ml culture. So what is the next step we do? We first do gram staining. I hope you all know about what is gram staining. This is the method by which you identify whether the bacteria is gram positive or gram negative, rod or cocci. So this is done microscopically, and then we identify whether your bacteria is gram positive or gram negative. So most of them, what we are dealing with is today gram negative rods. Except for Streptococcus, which is gram positive cocci, most of them are gram negative rods. So then we subject these bacteria to different biochemical tests called catalase test, oxidase test, indole, citrate, motility. Most of the Aeromonas are motile. And presence of fluorescence pigment, which is done for Pseudomonas fluorescens, potassium hydroxide hydrolysis. This is again another test which will tell you whether they are gram positive or gram negative. Gelatin. So if the bacteria secretes an enzyme called gelatinase, we look for gelatin liquefaction in the media, MRVP test, and glucose fermentation. So we do all these biochemical characteristics, and then we kind of shortlist that this could be Aeromonas, this could be Vibrio, this could be something else. So this does not give you a concrete picture. I mean, you cannot report just by doing biochemical characters that this is Aeromonas hydrophila and or whatever Pseudomonas. So we need to do further test to classify these organisms. Say, for example, I've just put a table where when you know that by gram staining, these are gram negative short rods. So both Aeromonas and Pseudomonas are gram negative short rods. And another important biochemical test is catalase and oxidase. Both Aeromonas and Pseudomonas are positive for enzyme catalase and oxidase. So we further shortlist that this could be catalase and oxidase positive, and other tests like indole, citrate, motility, hydrolysis, etc. are, their little motility, of course, is different from different Aeromonas. So, this is what we do just to come to a presumptive conclusion that these could be Aeromonas or Pseudomonas, but these are not confirmatory. Then what we also do is we extract the DNA from this organism and then we subject them to PCR. So normally we do PCR primers which are specific to the genus Aeromonas or specific to the genus Pseudomonas. I'm talking little closer to Aeromonas and Pseudomonas, but the same principle will apply if you're looking at other organisms like Streptococcus. We design primers which are specific for Streptococcus. Or if you're looking at Vibrio, we design primer which are specific for Vibrio, but not species specific. We go for genus specific primer at this stage. So we will amplify them using PCR. Here we use a gene called AeroM for identification of Aeromonas, and we use a gene called PAGS for identification of genes from Pseudomonas. And further, we either sequence the product which we have obtained from here, or we do what is called as 16S rRNA sequencing. So 16S rRNA is the part of the gene which is conserved in many of the bacteria. So when you do the sequencing, we get the idea like, this is almost certain that you are either having Hydrophila, Verunae, or Pseudomonas, or whatever bacteria. So if you know that what species you're handling, we go for the species or genus-specific primer and sequencing. But if you do not know what bacteria you're handling, then we go for 16S rRNA, which is a conserved sequence in most of the bacteria, and then we go for sequencing. So this is a flow chart which more or less works for many of the bacteria. So first I told you that once you inoculate the single bacteria into a liquid culture, we do gram staining. The moment we know that it is gram-negative rods, we subject it to oxidase test. If they are positive for oxidase, then it could be either Aeromonas, Pseudomonas, or Vibrio. So if it is negative, it could be other Enterobacteriaceae. So further, you subject them to glucose fermentation. So Vibrio produces acid in the presence of glucose and then turns the media color into yellow. The other bacteria, Vibrio and Aeromonas produces acid, whereas Pseudomonas does not produce acid. So the moment you have acid production in Vibrio and Aeromonas are confirmed, then we put them into a media which contains sodium. So Vibrio are halophilic. That means the Vibrio species can grow in the presence of sodium. Hence, we know that if it's growing in the presence of sodium, it's Vibrio. If it is not able to grow in the presence of salt, then it is Aeromonas. So further, you can classify Vibrio by molecular methods. Then once you know that it is Aeromonas, you can further either you can go for genus-specific PCR and then sequencing. That's what we normally do. Or if you want to do further biochemical analysis, we can go for esculin agar and glucose fermentation combined. So you have glucose positive production of acid and no production of gas, and esculin positive. Then we identify it as Aeromonas KVA. And production of acid from glucose, production of gas from glucose fermentation, and esculin positive is Aeromonas hydrophila, all three positive. Production of acid from glucose, production of gas from glucose, and esculin negative, we call it as Aeromonas sobria. So this is how we identify up to species level using the biochemical characters. So further, we will not stop with that. This is only presumptive. We can go for either PCR of the Aeromonas genus using primers which is specific for the Aeromonas genus, and then sequence them and to arrive at the final conclusion or what is the species level molecular identification. So this is how the species of Aeromonas is identified. So going to Pseudomonas. So I told you many of the Pseudomonas are fluorescent. So we look for the presence of fluorescence. So there are several species: Pseudomonas fluorescens, Putida, Pseudomonas aeruginosa, Pseudomonas syringae. All of them have fluorescence. Some of the other Pseudomonas are non-fluorescing, but we are very keen on Pseudomonas fluorescens and Putida in these species, and also Pseudomonas alkaligens. So here, once we know that it is fluorescent, we can further take the single colony and extract the DNA and either do Pseudomonas genus-specific PCR and sequencing or 16S rRNA gene and sequencing. Further, you will identify the species level, species of the Pseudomonas species. So this is the overall guideline for identification of different bacteria. Predominantly, Vibrio, Aeromonas, and Pseudomonas in the fish bacterial diseases. These are some of the PCR pictures where we have done the Aeromonas specific primers, and you can see these bands which are formed or because of the presence of Aeromonas in those bacteria. This is the genus specific PCR for Pseudomonas. So here we have used a primer called AeroM, and here we have used a primer called PAGS primer for Pseudomonas. And also, it is necessary for us to understand whether the strains what we are handling is virulent or not. So the Aeromonas infection has got multiple virulence factors, or many of the Aeromonas harbor a lot of virulence genes. Example, cell structural lipopolysaccharide, outer membrane protein, flagella, enzymes, and toxins. There are a lot of exotoxins which are very important for the pathogenicity of Aeromonas. Say, for example, aerolysin, hemolysin, cytolytic enterotoxin, and enzymes such as lipase, protease, nuclease. These are all very important for the pathogenesis of Aeromonas. Some of the Aeromonas are very, very toxic. It causes all the symptoms very fast, which could be because that they harbor all these virulent genes. So what we also do is, once we know that it is positive for Hydrophila or Veronae or KVA, we subject them to virulent PCR. Around 4 to 5 virulence genes are amplified to confirm whether this Aeromonas harbor any of the virulent genes. And these Aeromonas which are virulent are further taken for our management strategy, or we work for solutions which can kill this Aeromonas further. So in our laboratory, we collect the samples, we isolate the bacteria in selective agar, and then followed by enriched media. Then we do a lot of all the biochemical characteristics, which I discussed, like gram staining, catalase, oxidase, indole, citrate, motility, and fluorescent pigment, potassium hydroxide analysis. Once we know that, it is somewhere close to what we are looking at, we go for genus-specific PCR. If it amplifies by PCR, we go for sequencing. So once we know that we have the bacteria which is identified and typed, we preserve them by lyophilization. So now the second part of my talk is to manage these bacterial infection. So we all know very well that today we use antibiotics for the management of bacterial infections. So today we use multiple antibiotics. Say, for example, fish diseases are controlled more like TB. Multiple 4 to 5 antibiotics are dumped into the pond. This is resulting in multiple drug-resistant strain, which in turn enters into the human system. So that means most of the bacteria which are, there in the pond, in the fish, or in the shrimp, or in the poultry, enters into the human circulation, resulting in multiple drug-resistant bacteria in humans also. So that is the major problem going forward, which I'll come to it a little later. Also, many of these antibiotics are present as residues in the fish and in the other animals, which is posing a major problem if you're going to export your fish. So there is huge export rejection because of the presence of residues or antibiotic residues in the fishes. And also today, companies which were making huge number of antibiotics are drying up. There are limited number of antibiotics in the pipeline because earlier when they used to make antibiotic, the life of antibiotic for at least 10 to 15 years. That means They used to spend billions of dollars to develop one antibiotic, which used to be at least in the market for 10 to 15 years. Today, the life of antibiotic is just 1 to 2 years. So the companies are not able to recover the money they have invested in their R&D. Hence, we do not have newer antibiotic coming up very soon. So we are going to look at alternative strategies to control the bacterial infection in fishes. As I was telling you that the mortality, or what is the problem which is facing the world, is today AMR, antimicrobial resistance. So most of the bacteria which today we are handling are resistant to single or multiple antibiotic. So there is a projection which is reported in the review of antimicrobial resistance. By the year 2050, the death due to antimicrobial resistance is going to be major killer more than cancer. What is today, which is 700,000, is going to be 10 billion deaths, 10 million deaths by 2050. So unless we take very stringent action or use antibiotic judiciously, this is going to be a dire situation. So the authorities are very well aware of the situation, and they are making very strict guidelines for the use of antibiotics judiciously. So what I was discussing, we have plenty of bacterial problem. We have losses because of bacterial problem. Many of these bacteria are resistant to multiple antibiotics, and we have limited antibiotics which are getting developed. And we also have public health issues because of the antibiotic resistance. So we need alternative for antibiotics, which one of them could be bacteriophages. What are bacteriophages? Bacteriophages are obligate intracellular parasite that multiply inside a bacteria by using some or all of the host machinery. That means these are very small parasitic entities, biological entities, which can live only with the help of a bacteria. So they cannot at least multiply and survive independently. They need a bacteria to cling on and to develop and multiply. So these are the most common and diverse entities on the Earth biosphere. They are distributed wherever there is bacteria. So phages are there wherever there is high concentration of bacteria. They are present everywhere. It is estimated that for every bacteria on the Earth's surface, there is already 10 bacteriophages, which is naturally existing. So this is the nature's mechanism of controlling bacteria. So there are already 10 bacteriophages, which is existing for each species of bacteria, which is present on the Earth. So where do we find bacteriophages? Bacteriophages are present in the human and animal intestine. They are present in running water at a very high concentration between 10 power 4 to 10 power 8. So you see bacteriophages in the river, you see bacteriophages in the sewage, effluent. They are present in plenty in soil as well. So you can see almost around 10 to the power of 9 bacteriophages per gram of soil. So they are found everywhere in nature. So what is bacteriophage therapy? Bacteriophage therapy is the ability to use bacteriophages for the control of, bacteria. So it's an antibacterial agent, natural antibacterial, biological antibacterial agent. So this was first reported in the year 1896 by a scientist called Hankin. What he found was, in the river Ganges, people who were drinking water from Ganges were not affected by, there was a cholera outbreak in those areas in 1896. So people who were drinking and using water from Ganges were not getting cholera, but people who were away from Ganges were getting the cholera outbreak. So he took the water from the Ganges, he just filtered it and used it. It was still having the property. He heated the water, the property died. So he said that there is some living entity which is very minute, much smaller than bacteria because it is able to go through the filter, and which is temperature sensitive. That means it is living. So a living entity, which is much smaller than bacteria, is responsible for protection of these people who are having Vibrio, cholera. So this was the first reported evidence of bacteriophage therapy in the literature. So that means what I'm trying to tell is bacteriophage therapy is not recent one. It has been there for centuries. So how does the bacteriophage work to kill a bacteria? So this is the cycle. There are two ways by which a bacteriophage multiply. One is lytic cycle, another is lysogenic cycle. We only work on bacteriophages which are following a lytic cycle. Lysogenic bacteriophages are not suitable for therapy. So we select a bacteriophage which is lytic in nature. So this bacteriophage, as I told you, has got a head, a body, and a tail. These tail fibers have receptors, sorry, they have proteins which can go and specifically bind to the receptors on the surface of the bacteria. So the binding between a bacteriophage and that receptors on the bacteria is like a lock and key. So this goes, the key will go and fit only to the lock which it is intended to bind. So that means a bacteriophage against Aeromonas or Pseudomonas can kill only Aeromonas but not any other bacteria. So the first is it binds there and then it injects the DNA into the bacterial cell. Then it uses the machinery inside a bacteria and produces lots and lots of bacteriophages. Then, because of the pressure and the protein, which is called endolysin, which is released inside the bacteria, it bursts open, and then the bacteriophages, which is, say, it could be 10 to 100 in number, which is released and which again goes and attacks the other bacteria which is present in the water or in the animal. This cycle continues till the bacteria is present. The moment the bacteria clears, the phage cannot survive for a very long time and it also clears. So this is the way by which a bacteria or bacteriophage kills a bacteria. So let me just... So this was a video. Anyway, I'll come back to this video. I'm not able to. So this video explains the process, the same cycle which I explained to you now. The video depicted how a bacteriophage enters into a cell. It doesn't enter as a whole entity. It only injects the DNA and then multiplies inside a bacteria and destroys the bacteria. So now what are the advantages of bacteriophage therapy? These are self-replicating therapeutic. That means when there is bacteria, it replicates. Self-limiting, when the bacteria, if you're targeting Aeromonas or if you're targeting Pseudomonas, it, it will be there as long as there is bacteria and then it clears from the animal. So unlike an antibiotic, which will be there even after the bacteria is cleared, a phage gets cleared from the animal after the bacteria clears, which is a great advantage. So it's a green, natural alternative to antibiotics. And the best part of a bacteriophage is that it does not destroy the probiotics or the good bacteria which is present in the gut. So when you go to a doctor for some infection, if he prescribes you an antibiotic, you're going to lose all the good bacteria. So he gives you a probiotic along with that. Correct? So that, your natural flora is getting recouped. Whereas when you give a bacteriophage, it'll hunt only the pathogenic bacteria or the target. So we always say antibiotic is like a nuclear bomb. Phage is like a targeted missile. It will go and attack only the bad bacteria. So it is because it is highly host specific. So what are the other methods of bacterial disease management and how does it compare with the bacteriophage? So we can also manage disease with probiotics. Of course, probiotics have non-specific action. So it can, compete with the pathogenic bacteria for the binding sites in the gut, or it can produce, and by competitive inhibition, it can compete for the nutrients in the animal or in the system, or it can produce something called bacteriocins, which can in turn, kill the bacteria. So these are the ways by which a probiotic works, but it is, it can be a good synergistic action with phages. Uh, but again, it cannot have a very specific role against bacteria. Now we also use essential oils, which have a very good antimicrobial spectrum. Vaccines, of course, are there. I mean, fish vaccines are also coming up against Pseudomonas hydrophila and so on. Organic acids have been used with some success. Antibiotics, of course, are being used. And next is bacteriophages. So if you see all these scenario with different aspects like application scope, treatment cost, weak points, reliability, ease of implementation, and amount of actors in the market. So we see phages are good in many of the aspects. All the good aspects, phages form a big circle when compared to the other treatment strategies. So, and what we did, we have done extensive work on Aeromonas hydrophila and Pseudomonas fluorescens. So we have developed phages against Aeromonas hydrophila. And then we also do testing. So this is one of the challenge studies where we have taken around 210 rohu in 7 different tanks. And then in the first set of tanks, we have immersed them with Aeromonas hydrophila at a concentration of 3 into 10 per 10 CFU per tank. In the treatment tank, we infect them with Aeromonas hydrophila and also fed them with bacteriophages. In the control tank, there is no Aeromonas and there is no phage. So in one tank, it is only Aeromonas. In the other set of tanks, there are Aeromonas along with the Aeromonas hydrophila specific phages. So let's see how the results are. So you can see that the survival in these tanks, average survival of the 3 tanks, when you have treated with phages, after 15 days, the survival was around 96.6%. Whereas when you have given only Aeromonas without phages, the survival after 15 days was only 50%. So you can see from day 7 onwards, there is low mortality and it is only 50% after 15 days of challenge. So we also see, I told you, this is a specific agar which grows only Aeromonas. So we can also see that there is reduction. So the one which is there in the left side is the control plate. And the one which is on the right side is the treated plate. So you can see that both of them, this is on day 6. So you can see there is slight reduction in Aeromonas in the treatment. And day 7, there is further reduction. And, day 9, I think it's not coming. This is day 9. So you can see that the Aeromonas are almost cleared from the animal. So this is the product which is called AeroShield, which is the phage cocktail of Aeromonas and Pseudomonas. So this is being used for treatment of Aeromonas and Pseudomonas. So this is another challenge studies, which is done externally in a government institute. So we have set of tanks where we have only Aeromonas, and we have 2 sets of treatment tanks where we have Aeromonas phages along with the Aeromonas host. And in the control tank, there is no Aeromonas and no phages. So each tank contained 30 animals each, and these are all done in duplicates. So we can see that the mortality in only Aeromonas tank, when there is no phages, cumulatively is around 100% after 66 hours of infection. I mean, slowly it is coming. After 30 hours, we have lost most of the animals. And in the treatment tank, it was survival is almost 78%. The mortality is 16.6 and 26.6. In the other tank, it is 6.6 and 26.6. So the survival, the average survival in the only Aeromonas tank is only 0. Whereas in the other two tank, it is around 78 and 83% survival. Control tank, there is no, sorry, this is 100%, this is a mistake. Control tank, there is no death. So this is in short about how Aeromonas, I mean, how we identify Aeromonas and other organisms from the diseased animal, and how we develop a strategy. Today, we are using antibiotics to curtail the infection of these, but how we can use natural and the plausible alternative to antibiotic, which is bacteriophages, which can be used to control infection. And we have done n number of studies, and this is being used in the field also to control Aeromonas and Pseudomonas infection. So with this, if there is any question, I can take up. Thank you.
Yeah, thank you very much, madam. Very detailed presentation.
Thank you.
I request the participants to unmute yourself if you have any doubts, or else you can also type in the chat box. So let me ask the first question, madam. Now
Yes.
we are talking about the known pathogens.
Yes.
For example, all of a sudden there is a new outbreak, with the new symptoms. How do you proceed?
Okay. See, there is a possibility called metagenomics. Okay,
Okay.
today metagenomic study is also there, which I have not spoken with. Yes, in human and other places it is popular, but when we try to do metagenomics, what we are getting is a lot of probiotics, because probiotics have been used extensively in fishes and shrimp. So metagenomics is one way by which we can do. And second is that we can, plate and select the colonies which are large in number. So we cannot be doing every sequencing. So you can take enrichment media like LB agar or nutrient agar and pick up the single colonies, inoculate them individually. You can do biochemical analysis and you can go individual colony 16S rRNA sequencing. So that is one possibility, or metagenomics is second possibility.
Okay.
Madam, is it audible, madam?
Yes.
Instead of going for biochemical tests and any other tests to identify the bacteria, by seeing the fish itself, can we identify that by particular bacteria is attacked to this fish?
Okay, not always it is possible. Not always it is possible, but yes, farmers are to a certain extent go by the symptoms like red disease, red sore, see, it is certain diseases where the symptoms are established, like, red ulcers and red disease. Yes, then we can boil down to Aeromonas or Pseudomonas. But if there is a new outbreak, or if you don't even know what are the new symptoms, then we may have to go by the other method, biochemical and molecular methods.
Madam, farmers will be asking the symptoms and they will be explaining symptoms and they will ask for what is the disease.
Yes, yes. Most of the time, yes, established symptoms, and then because we cannot be sequencing, this is in case, more of research where we want to develop a solution to the problem. We need to be very certain about what bacteria we are handling. So we go for all these methods by which we identify and isolate. But it is impossible that every case you take it, isolate, and then give the treatment strategy. No. So it is, so when you go to a doctor, he always doesn't ask you to go for antibiogram or sensitivity. Looking at the symptoms, he gives you antibiotic. So that's how we also have to strategize. We know that these are the symptoms in Aeromonas, these are the symptoms in Pseudomonas, and other diseases like Flavobacterium, which is not really established very well. So those cases probably we may have to select few cases and go for identification, but more or less we go by the symptoms.
Thank you, madam.
So any other questions from students?
Good afternoon, madam.
Good afternoon.
Madam, how we can isolate the bacteria phase?
Sorry, I'm not able to hear you.
How we can isolate the bacteria phase?
How we isolate the bacteriophages. Bacteriophages, as I told you, are present wherever there is bacteria. So we normally isolate from sources such as sewage or say infected, highly infected areas or highly fish contaminated area. Those kind of places we select, sometimes oysters, so the different places we collect samples, and then we trap them with the host. Say we have Aeromonas hydrophila or VM. So we have specific target. So we try to trap these phages using the host which we are targeted to kill. And then we go and purify these phages and then characterize further.
Is there any specific media for isolation of bacteriophages?
Media for bacteriophages? No. Bacteriophages are, they cannot grow on any media. They grow only inside a bacteria. So first you have to grow a bacteria and then grow the bacteriophages inside a bacteria. So there is no specific media for bacteriophages.
Okay.
Yeah. They don't grow in the media, they grow inside a bacteria. So whatever media in which a bacteria grows, this will grow inside that.
Okay, thank you. Ma'am, at what concentration of bacteriophages we have used for challenge studies?
See here, I think 10 power 9 CFU per gram was. So you can see that per tank, I think these are around 15 liters or 20 liter tanks. So I need to reconfirm that. So we have used 3 into 10 power 10 CFU per tank. So normally we go, in this challenge study, we at least use 1 MOI, that is called 1 multiplicity of infection. Every bacteria, that is 1 bacteriophage. So that is the level which we have added. So that is the level with which we have added here. So normally we use anywhere from 0.1 to 1 MOI under the field conditions for treatment.